Hey guys, today's topic is blastulation, which is one of the stages of early embryogenesis, which is characterized by cellular differentiation. So now let's get back to fertilization, where we have the ovum or the egg of the female waiting inside the fallopian tube. Okay, the egg has two layers around it, and the inner layer is called zona pellucida, and the outer layer is called corona radiata. Now here comes the sperm, and once the sperm fuses with the nucleus of the egg, the outer layer corona radiata is going to disintegrate, or it's going to wear off, leaving behind only the zona pellucida with a special type of cell called zygote inside of it. So this is the zygote. The zygote has two N chromosomes, which means it has two sets of chromosomes, one from the mother and one from the father. Now this zygote is going to start dividing by mitotic cell division, and it's going to undergo cleavage around the 24th half hour or so. So in cleavage, basically the zygote is going to split into two within the zona pellucida. So one zygote, that single cell zygote, is going to divide into two cells inside the zona pellucida. So there's not going to be any increase in cell size. Now this mitosis is going to carry on, and the cell keeps dividing and dividing, all the while remaining within the zona pellucida. Okay, so it's going to reach the 4-cell stage, and it's going to reach the 8-cell stage with no increase in cell size, because everything is taking place inside the zona pellucida. Now on the mark of the 3rd day or so, the zygote is going to enter into a 64-cell stage, which is called the morula. So everything inside the zona pellucida still, and this entire structure containing 64 cells, is called the morula. Now if you see, each and every single cell until the morula stage is exactly identical to each other in terms of structure and function, and every single cell here up to the morula stage is called a blastomere. Okay, so the word blastomere comes from a word called blastos, meaning spores. So basically what's happening is every single cell is producing a spore of itself, and the cells are dividing kind of like that, and it's called a blastomere. Now on the mark of 5th day of the embryonic development, we're gonna see blastulation. Okay, so blastulation is a significant event because only from blastulation the cells are going to start differentiating. So the blastomeres are going to develop into two different types of cells in the process called blastulation. Let's see how. We're still going to have the zona pellucida, no increase in cell size. Okay, we're going to get a structure called as blastocyst. Okay, now let's see what a blastocyst is. It's a hollow sphere of cells, and the outer layer of the blastocyst is going to contain a different cell population, and the inner part of the blastocyst is going to contain a completely different cell population, and also it's going to have a fluid-filled cavity inside. This entire spherical structure, containing one different cell population on the outer part, one different cell population in the inner part, with a fluid-filled cavity, is called as a blastocyst. The cells in the outer part of the blastocyst are called trophoblasts. And the fluid-filled cavity is called blastocoele. And the cells in the inner part, or the most inner part of the blastocyst, are called as the inner cell mass. It's abbreviated as ICM. Now, let's see what is the difference between a blastomere and any cell inside the blastocyst, okay? So we have two different cell population. We have the blastomeres and the blastocyst cells. Blastomeres are totipotent in nature, which means one single cell is enough to give rise to an entire organism. This principle is usually used in IVF or in vitro fertilization, where they harvest any single cell from the 8-cell stage of the embryo, and they culture it in vivo. And once the 8-cell stage develops into a blastocyst, they're going to implant it into the uterus of a healthy female for development into a complete baby. Okay? Whereas if you take a blastocyst cells, they're not totipotent. They are not totipotent. They cannot give rise to an entire organism on their own. They are multipotent though, which means they can give rise to many different types of cell lines, but not the entire organism itself. Now let's see the specific functions of the cells inside the blastocyst. Okay, we have the inner cell mass cells and the trophoblast cells, right? So the inner cell mass cells give rise to 3 derm layers of the body, so which are the ectoderm, mesoderm, and endoderm. Now ectoderm, give rise to the cells which are basically absorbed externally from the body. So we can take skin as example, okay, and lens, nails, hair, and other things which we can see from outside of the body. And it also gives rise to the development of the nervous system. Now mesoderms is it's seen in the middle of the body. Meso means middle, so let's go a little into the middle of the body. So it's going to give rise to things like the skeletal system, muscular system, and so on. And the endoderm is in the most internal part of the body. So we can take the internal organs as an example. So we can say kidneys, liver, etc. Okay?